can remember when I was trying to explain to my mother epigenomics, and she just shook her head and said, now I'm going to get blamed for everything that happens to my grandchildren. And she said, it used to be you are what you eat, and now she said it's going to be you are what your grandmother ate. So she was very concerned. So thank you all very much. And I think I might just take a prerogative here and start off with a question. So organizations like Genome Canada and Genome BC, one of the things we do, we develop funding programs. And I think that the programs that you participated in came about in different ways. And I think in the case of the Forge project, it was really a project looking for a funding opportunity, looking for a program. It sort of was out there just waiting for that. And that's why I think, as you said, Jan, it came together so quickly. It was just well orchestrated because there had been that collection of samples. The clinicians had been really primed, the pediatricians, and so I think that was an interesting mechanism that we were looking for an opportunity to really demonstrate how next-gen sequencing, as you said, could have application quickly. And so it was a perfect fit. And now the other two programs really came about because of these international initiatives that were ongoing, and CIHR and Genome BC, and in the case of the Epigenome, Genome Quebec came together to create a funding opportunity to allow Canadians to be part of that. So what do you think are words of advice to funding agencies like us, how we can be really on top of and aware of the types of programs that we need to ensure that we create across the full spectrum? As I said, Genome Canada, we will continue to fund discovery, apply, translational research, the same with Genome BC. Any thoughts on that? Jen?
I think there were really two things that came together for Forge. One was the clinicians and the samples and the patients that were available, but the other was the technology. It had gotten to a point where this really was possible. And I think the community recognized this. We weren't the first to do this. Certainly there were major efforts in the United States and Europe, and there continue to be major efforts in the United States and Europe to do this. But I think it's just sort of a question of keeping in touch with the people who are on the ground and see the problems and are aware of the advances in knowledge and technology that enable us to have an opportunity.
Martin and Deborah?
Yeah, I mean, I would agree that consultation is key. I mean, in the case of the Epigenome project, really there was a champion as well within the funding agencies, Tony Phillips and others, really driving that agenda forward. I think having that champion there who had a vision to bring it to the fore, I think was very important. But I mean, that came through consultations, there were meetings, there were workshops that CHR put on around the epigenome to look at feasibility and interest within the Canadian. So I think those type of activities, I think, are very important.
I guess I would really just echo that. It's that sort of constant communication with the research community. In our case, in particular, we were also assisted by, early on, Michael Smith's seed funding for just a little piece of our team, and then CHR and Genome BC and others, just really kind of being there at the right time when the various Canadian teams were just about kind of that launch level, and then having the funding ready. So, I mean, it's all about timing and kind of reading both the global and the national situation well. But if Canada's going to really stay, at least as a contributor, I think that's the kind of thing, these targeted approaches is really vital.
And it's great to see that there are such great teams that were able to step up to the plate and make sure those programs are successful. So thank you. So I'm going to open it up now to questions. I think we had 3 great talks this afternoon and real examples of applied research. So open the floor. I'm hungry. So I
So I had a question for you, Dr. Money. There's a lot of sort of anecdotal evidence. I've heard that women who have daughters as preterm then go on. Those daughters have preterm babies as well. And have you looked at, or have you thought about looking at any correlation in that?
There is some evidence that there is multigenerational prematurity that doesn't appear to be kind of classically genetic, and we are concerned that there may be this altered microbiome. We had actually originally in our proposal had a multigenerational plan that we actually felt was going to be too complicated to try and fund in our time zone, but that is something that our team, members of our team, are really very interested in understanding that microbiome transition. Obviously, there's probably other factors other than that, but it is very much a question.
I was thinking more in terms of also the epigenomic.
Yeah, because that, if I may, my question was going to be, what's your thoughts on the microbiome's effect on the epigenome?
Oh, I mean, I think that's a very fascinating area, certainly within immunology, trying to understand the gut epithelium and how it's reacting from an epigenetic perspective to the microbiome. And I'm sure that's true also in the vaginal epithelium. So I think but you're taking two very complex areas with metagenomics and epigenomics and merging those. And I think the technology is getting there, the analysis techniques are coming together, OTUs, etc. And I think it's really an emerging area, for sure. Yeah.
Mohammed, did you want to follow up, or is that's okay? Other questions? So Deborah, I have one for you. What is your link in your engagement actually with the International Human Microbiome Consortium? How do you interact with them and engage with them?
I have to be honest, it's really more at the sort of conference level, collegial level. At least our team hasn't had a formal collaboration with other teams. There aren't that many teams actually in our particular niche area. We have a relationship with some of our colleagues in the United States just because we know them better geographically near, but we haven't been as integrated with the international efforts as I think some of the GI groups to say.
Thank you. Brad.
Jen, can I just ask, in regards to the Forge approach, kind of using next-gen, in regards to using kind of next-gen sequencing as an approach to evaluating some of these kids with, tough to sort out conditions. So can you just talk a little bit about the scalability of that? I believe there's some groups that are trying this clinically now. Is it working? What do you think about that? Is this still research, or are we at the point where we can start to push the envelope and put it into more clinical practice? What's necessary to get us to that point?
So it sort of depends on what condition you're looking at. Yes, there are a number of programs throughout North America and Europe that are now offering sequencing, exome sequencing in particular, clinically, through clinically approved and accredited laboratories. The only one that exists in Canada, to my knowledge, has recently been set up at the Hospital for Sick Kids, but there are probably 8 or 10 in the U.S. There are also a number of companies now that offer whole exome sequencing as for anybody who wants to pay for it. So it is. We have used it clinically in maybe 6 or 8 families in British Columbia, funded through our medical services plan on a one-off basis, in special cases. So there are circumstances in which I think it's unequivocally sensible to take this approach. And these are usually circumstances not so much intended at gene discovery, but intended to find which of several possible genes cause a particular condition, when knowing that is important for treatment. So that's sort of the lowest low-hanging fruit. And those are cases where in some instances we've been able to get funding for clinical sequencing here. It's being done in many places throughout North America and Europe. In terms of it being routinely available, and in terms of its application to less low-hanging fruit, to more complicated conditions like intellectual disability or autism, for example, I think those are still research applications. We still need to know more in terms of what the range of genes that can cause these conditions are. Intellectual disability, there's probably something like 1,000 different genes that could be implicated. So exome or possibly whole genome sequencing make a lot of sense once we know what all of those are, the vast majority of those genes are. Because when you're working clinically, you're usually not looking to discover a new gene, you're looking to discover something that you know so that you can intervene. So we're sort of in the early steps of clinical application.
More questions? Brad?
So sorry, if I can ask a follow-up, and maybe I can ask it to all of you though. So we talked about the user partnership program this morning and about the GAP program. And so we're really looking for the end user, or in this case, it would be the PHSA or one of our health authorities to actually, Vancouver Coast or whatever, to be driving the actual desire to want to move this technology forward. So Deb, maybe in your case, in the case of prematurity, so the tests that you're developing, I mean, to me it would seem that that could be incredibly valuable. And Jen, same thing for, looking at again, these small number of patients that are really difficult cases. What do you believe it would take to get them to partner on something like this to actually help you drive your technology forward? And because you could look at them as the true end user. I mean, Deb, in your case, you could also look at potential partners from the diagnostic industry that would be interested in this, and potentially even Jen in your case. And Martin, I think the same thing applies. It's maybe just a step out, but with some of the epigenetic applications that we could think about. So maybe I'll just open it up to all of you, that question.
I think in our case, prematurity is kind of the holy grail, the enormous cost to the healthcare system, which I hate to say is what health authorities are thinking about, to having nurseries overfilled with extremely premature, very ill children, and also the consequential societal and medical costs of these children really lifelong. If we could even reduce that by some significant proportion with a screening and/or a test that either screened and/or screened and provided opportunity for preventative treatment, I mean, that would be astounding. We're not there yet. We believe we are getting there, but I think we're clearly going to have some, proof of principle clinical trials to convince health authority level funding,
I think it probably will take more than that. This is not an issue that I think is more relevant to any particular health authority in BC than it is to the Canadian healthcare system in general. But because of the way our healthcare system works, it tends to be kind of risk averse and not. And in BC, certainly, there's almost a brick wall between research and clinical care, and it makes it very difficult to be out front, kind of innovative. There's an opportunity once it's been established, maybe through these research projects or through other places in the world, that you've got something that will save money and does provide better care. Then I think it becomes an opportunity. But it's difficult in our system to justify being the first adopters. It's just the way that we're organized, that we don't. There isn't a clear-cut portion in the Canadian healthcare system, in the service provision side of it, that sees innovation as a major area for investment.
So I mean, from an epigenetic perspective, I would argue that we're there now. I think in terms of making the case, I would argue that it's about money at the end of the day. So that project needs to be set up to convince PHSA that it's actually financially in their best interest to actually move forward with a certain technology. Let's talk about, for example, BRCA1 screening. So BRCA1 screening, which is done under the clinical hereditary cancer screening program within BCCA, which is actually done on an NGS program now, NGS platform through the CAP-accredited platform at the Genome Sciences Center. But we know that there's a class of women who have early onset breast cancer that is due to DNA methylation in the promoter region of the BRCA1 gene. So we're looking for the genetic lesions, but we're completely ignoring the epigenetic lesions, even though we know they're there, and the technology exists to capture them with the same efficiency that we capture genetic lesions. So I would say putting together a program where we could prove that this is doable and it's going to save the PHSA or the province money would be the way I would approach it.
There's a question at the back.
I had a question for Dr. Hirst. But you mentioned that epigenetics can be, the effects are from grandparents to children. It could be even beyond that. It has probably not been checked. But what is the method in place to control for variation you will see in your test subjects in terms of data analyses, and how do you control?
Well, so the studies that have been done to date are epidemiologically. So these are correlative studies, right? So I mean, there are a lot of confounding factors. In terms of hard evidence, that's really come through model systems, such as the work of Mike Skinner and others who have shown out to the F3 generation in a rat model that exposure to a certain pesticide gives an increased incidence of certain types of tumors out to the F3 generation. So that is true transgenerational inheritance because, of course, the F0 is present in the pregnant dam, and then the F1, the F2, and the F3. So I think right now, in terms of transgenerational inheritance, that's the hard evidence is in model systems. Our understanding of it within human populations is pretty correlative at this point. Yeah.
Other questions? In the back. Brad, put your hand up.
Hi, I'm interested in the effect of pharmacotherapy on the work you're doing. Because when you work on the genome, it's one thing, but when you start working on the microbiome or even the epigenome, it's a different thing. So my question, maybe to start with Dr. Manis, is how do you monitor pharmacotherapy? And I'm thinking of your HIV population, for example. Presumably these women are treated with various cocktails of highly active retroviral therapy. Adherence is an issue. How do you monitor, how do you control that, and how do you take that into account?
Excellent question and complex situation. Certainly in our single time point cross-sectional studies in our HIV population, we're not pretending to be able to answer that. We are at least getting a hint of the effect of that by having the ability to know the entire antiretroviral history, the HIV variant that the person's infected with, the particular actual genotype and phenotype of that virus in that person, and their immune status at the time of sampling. So we can get at least some hypothesis generating results from that kind of study. But certainly for us to understand the real impact of pharmacotherapy on the microbiome, it's going to require much larger scale multi-sampling studies over time, looking at on and off drugs to try and show that. That clearly needs to happen. We're just not quite positioned to do that yet.
Hey, me again. I have a general comment with you guys out there. Somebody with a who's the father of a 9-year-old who's a preemie and somebody who hangs around with Martin Hirst a lot, I think we should have a major epigenomics study of preemies in general, because there's certainly they have lots of fascinating phenotypes. My question is actually much more trivial than that for Deborah. When you do we're quite interested with the next-gen platforms in doing 16S or other microbiome sequencing, like on MySeq, for example. What read lengths do you actually need? Do you know sort of what the target is? Because we've been extending them out, but we've done some initial experiments which haven't been totally satisfactory.
Well, I guess it depends on sort of level of discrimination that you want, and so shorter read lengths, you're going to get less detailed information. We chose CPN 60 because of our ability for discrimination, and our read lengths are longer. But you know, I think it depends on whether you're just kind of trying to get a large scale, you know, things like the gut, where the incredible number of organisms and gene level, genus level data is sufficient. Read lengths can be quite short, and you can get just kind of a survey sense. If you're trying to look at fine shifts in a microbiome, perhaps related to drug therapy, I think you have to look at it differently. So it really depends on your purpose, I guess.
Roger.
Deborah, on your chart of potential outcomes, you sort of say one of them is to be able to identify the likelihood of preterm birth. The question is, what would you do about the answer? If you, in fact, identify a much higher likelihood of preterm birth, what do you do about that?
We presume that we're going to be able to alter the microbiome in a positive way. So a lot of the studies that were done pre-metagenomics were targeting women with abnormal microbiome, defined as bacterial vaginosis, and using sort of broad spectrum antimicrobials to sort of blast that microbiome and try to shove it over to a more positive situation. The trouble is that the results of these broad spectrum antibiotics are actually to cause as much disruption as you had at the beginning often. So I think what we would hope is when we understand the right microbiome clusters associated with successful reproductive outcome, and those probably more than one associated with prematurity, preterm rupture of membranes, then either preconception or in the first trimester, we would be using more targeted antimicrobials and or probiotics to just nudge the microbiome back to a healthier environment in the hope that you did it early enough that you could get a positive outcome. So there'll be time windows and subtlety to that. So that's a number of studies down the road, but we have them at least notionally in our heads around what we would want to be doing.
So you feel that if that was almost a standard test, you'd be able to identify that potential quite early in the pregnancy, no?
Or even preconception.
Okay, good.
More questions before Brad? Anybody else? Did you have one, Brad?
I do.
So, you know, you're all three to be commended on the great work that you're doing. I mean, these are just fascinating to hear the updates. Even though I feel like I know these projects fairly well, it's just great to hear the updates and see what's going on. So these are applied research projects, but they clearly have great translational potential. I mean, all three of them. And I guess the question really is do you have those next steps mapped of what you need to do? Does Genome BC, does Genome Canada, or do other programs exist in Canada to help you do that next step? And you know, is there something else the Genome family in Canada here can do? You're speaking to both Genome BC and the Genome Canada. Is there something else that we should be, again, it's back to Cindy's first question, that we should be doing for you? Because I think that what we would hate to see is that you guys hit a cliff and these projects just, you know, don't have the runway that's necessary to take them to the next level. So maybe to each of you, just to ask you to kind of just comment on that.
Well, I can say that I think the availability of funding for innovation, these translational kinds of innovation is absolutely key because of the limitations that we do have in the funding through the healthcare system, and also on the CIHR side, where sufficient amounts of money to do really translational things to get work all the way into the clinic is very difficult to come by. So in the area of genomics, we're fortunate to have Genome Canada and the various provincial genome centers to exploit the tremendous technology as it becomes available, and the tremendous advances in knowledge as they become available. But there is an absolutely key role in appropriate competitive research opportunities, and particularly ones that, like the one that I think we exploited with Forge BC, have a low activation energy in terms of being able to get into the system.
So I think I would echo all of Dr. Friedman's comments. I think one of the issues, at least in genomics and epigenomics, is the issue of the data. So currently, within our project anyway, when we generate a reference epigenome, there's nowhere in Canada to put that. We cannot store it anywhere. That's true of all of the genomes that we generate, human genomes that we generate at the Genome Center, and of course it's true across Canada. Right now we submit our material to the EGA, which is a tiered access protected database, but really there's nowhere in Canada to do that. And I think that is a fundamental infrastructure that, if you look out into the future, is something that Canada should really be considering building to really allow for translation.
Let me just comment on that, because you're absolutely right, and it sort of illustrates one of the problems that we have. If we want to, for example, do clinical sequencing within the context of a health authority, the information technology and the storage capacity is just, it's completely not adequate. So it's a huge thing. But whether the right approach is to fund something like that through Genome Canada, if it's for clinical service, I think that's open to debate, but it could be that Genome Canada and Genome BC could have a very important role to play in terms of catalyzing those changes and improving the infrastructure that's necessary. It's probably more money to do it right and to make it available throughout the country than Genome Canada or the provincial genomes have. But there could be a really important advocacy and catalytic role.
So, I guess I would echo some of the comments. Certainly, bioinformatics coordination across the country and the support for that, I think, could improve all of the projects. From a microbiome perspective, we're kind of the sort of slightly poor cousin to the major genome initiatives, and certainly, as is suggested by some of the questions this afternoon, Next Steps is going to need multiple sequential sampling and studies with interventions and studying different diagnostic platforms, studying different types of interventions, and that's going to require a whole series of funding calls, and currently at the moment there isn't anything really that fits that, because we're not quite at the kind of call that you were speaking of this morning. So I think there is a risk that projects like ourselves and other Canadian microbiome projects could hit a wall, because there's still significant translational research that needs to happen before it hits prime time. So I hope that those opportunities appear.
So it's good to hear data, bioinformatics come up, because I think that we were just at the plant and animal genome meetings in San Diego, and I don't think I went into one session. They were talking about their new strategy for plant genomics. It was all about data and bioinformatics. 80 to 90% of the problems that they perceive going forward is that. And so, you know, we are in Ottawa, the federal funding agencies have come together to try to start thinking about how to deal with big data in Canada. And there's a lot of initiatives going on, digital data and all of that. We're trying to really understand and get our heads around that. So right now it's NRC, CFI, CIHR, and NSERC, and SHRC is involved as well. And I think that it is something that we really do have to tackle. In addition to, you know, the standalone bioinformatics and computational biology funding opportunities, we need to have a national plan.
In all of the discussion that's gone on so far, I've heard a lot about public funding agencies and hitting the wall with getting help through federal or provincial sources. But I haven't really heard anything about how pharma plays into all of this. Like, pharma has a lot of orphan disease research and development programs. They do a lot of research in probiotics. So can you comment on how they might play into all of this?
I mean, I think they have a potential role, but it was a casual conversation earlier. We were talking about this. You know, the challenge is when you're trying to really answer some broad-based questions, pharma has very specific purposes. You know, they want to either develop a diagnostic or a therapeutic, and you really have to happen to have the right match to move your research agenda forward in partnership with pharma. And in Canada, we don't quite have the same volume of companies with large pockets as other countries do. So I think it's just, if it works out, it's brilliant, but it isn't necessarily the solution, I don't think, for each research project.
I would agree with that. I think many of the conditions that we deal with are rare and are only, they tend only to be of interest if pharma sees an opportunity to generalize on some of the pathways, for example, that we may be involved in. And that tends to take a fair amount of work before you get to that point. So it's a bit after the diagnostic stuff. On the diagnostic side, there's a lot of interest in the States, certainly, and but in the Canadian perspective, we want to sort of try to avoid being priced out of the market because those tends to be an expensive way to go. So we tend to try to develop ways to do this that are fundable within our routine healthcare system.
Yeah, I mean, I concur with the comments. I think, you know, pharma is certainly downloading R&D activities from, you know, their internal operations to the academic sphere, and I think those opportunities exist. You know, it's about making connections and networking. They certainly exist within the epigenetic community, and there are a number of large pharmaceutical companies who are interested in developing those therapeutics. So I think they exist. I've spoken to pharma that have come by. What are you doing? Is there something we could be interested in? You know, so I think it's a one-on-one networking to try and find that right fit. Yeah.
And I think that pharma is looking for new ways to do their research and looking more for academic partnerships, public-private partnerships. And I think our two programs, GAP and UP, that you heard about today, one of the hopes is that we will attract some pharma interest in actually partnering with academics. I think that's time up. Okay, well, thank you very much. Great.